So let's get started. So today's, for the next sort of 20 minutes or so, I will sort of explain what single cell analysis is all about, what are the different things, requirements you need to keep in mind when you're embarking on such a project, what are the bottlenecks you could face, and I will kind of explain how Singleron's end-to-end workflow can help you with this. So to just start with a quick intro about our company, Singleron. So we do want to focus on providing end-to-end accessible single-cell multi-omic analysis to our customers. So we are present worldwide in the US, Europe, and I am from the Singapore office. I oversee the regional APAC tech support. And so we are based in Singapore. So to just start with single-cell sequencing, we take one step back and talk about bulk RNA sequencing. Maybe that's what people are most familiar with. And so if you kind of think about a fruit bowl with a lot of different kinds of fruits, what bulk RNA sequencing helped you do was kind of make a smoothie out of it, and then you get all your results back. And then what you do get from that is very useful information about a sample, about your bowl, but it's an average profile of the entire bowl. So you kind of have an idea that there's all of these fruits mixed, but you cannot get an idea on what exact fruits are or what the number of each fruit is. And so single cell sequencing kind of gives you that information. So you take a fruit bowl, but this time you're individually assessing every piece in it. And so your results correlate to each kind of piece you took out. And so you have precise profiles. And so this is very useful and important. So moving from a fruit bowl example to an actual biological question. So if you've heard about a tumor, it has a microenvironment, and we all know that there are many different cell types in such a tumor. You have the actual cancer cells, you have immune cells circulating within this tumor, stromal connective cells, blood vessels, other cell types such as fats or neurons, and of course cells that are non-cancerous within this tumor. So to get an understanding of all these different cells, if you actually did a bulk sequencing, so you take this entire tumor, mash it up, and run a bulk RNA-seq, you get an average profile. So if you look at the different genes that this sample has, you will see that, say, gene 1, it's very hard to tell if it was upregulated or downregulated. Gene 2 in this sample, you would assume is upregulated, and gene 3 is downregulated. So this is kind of information, useful information that you would get from bulk-seq. But say you had an option of actually finding a way to partition the different cell types and then look at the gene profiles. And so here you immediately understand that gene 1 is upregulated in certain cell types, but downregulated in other, so on and so forth. So what single cell helps you capture is capture like the different cell types that are in the sample and their interactions and interplay. And so if you think of just all organisms and tissues of interest, all of them contain a high diversity of cell types. So there are so many different questions that remain unanswered when you do bulk RNA-seq, but can be answered once you decide to embark on single cell RNA sequencing. And so it's not surprising that many different fields now have studies that have utilized single-cell sequencing to answer very important questions. So I've just picked a few publications just from this year. So you can see there's studies that do single-cell sequencing to build an entire atlas. So then you know different cell types that probably are novel. Nobody knew they existed in these different tumors or cancers. And also you identify rare cell types that are very important probably to find out as biomarkers or therapeutic targets. Another field is immunology. There is a lot of single-cell sequencing happening here when you're interested in understanding immune diseases and cellular responses. Developmental biology, how do different cell types develop in aging, what are the different genes, gene expressions, etc. Drug development, so if you're interested in actually identifying biomarkers or targets, you've got to do single-cell sequencing now to identify these. And of course, most commonly used in clinical or human samples or mouse models, but now single-cell has actually moved to various different organisms and species. So plants, zebrafish, other non-human primates, etc. So single cell is now applicable on all different species as well. So for this webinar, I will kind of cover these 3 points across the different topics. So one is, how do you get started and set up single cell analysis? Then what are the considerations and bottlenecks that you might face and how you can overcome them? And then at every point, I will also kind of mention what Singleron can offer for each of these steps, so you can consider that for your experiments. So what you see here is a very standard workflow for single cell or single nuclei. You start at getting your tissue, preserving it, dissociating it, finding a way to partition and capture the mRNA. You have to sequence it, and then finally, you do the bioinformatics. And so I also want to just point out that Singleron has products on all of these steps, and so we can really go end to end for you. So you've decided to get started with single cell project. What you first need, of course, is your tissue. So if you want to do single cell work, you kind of have to have fresh tissues. It can be organoids, spheroids, etc. Or you need to have cryopreserved cells. Example is PBMC. So if these are your tissue types, you can go with this workflow. If it's a tissue, you need to kind of preserve and dissociate it, and then you go down the workflow. If it's cryopreserved cells, then you don't need to do any dissociation. You directly load the samples and carry on with the workflow. Now, in case you have flash frozen samples, you can still embark on doing single cell, but here it will be single nuclei. So here you take your tissue and isolate your nuclei before you actually go down the workflow. So I will do a little bit more of detail in this later. So starting up, you need a way to preserve your tissue, especially if it's a surgical biopsy sample or you're actually dissecting out from a model organism. It's important to have some way to preserve it, either for storage or transport to your lab. And the most important thing to keep in mind is the tissue needs to be maintained with high viability of cells. So what you can use for that is our tissue preservation solution. So this is sort of a solution that will stimulate the physiological environment and maintain high viability. Our product is able to keep your sample high viability in your sample for up to 72 hours at 2 to 8 degrees. So you get about 2 days to 3 days to actually transport or store your sample. And this is some data that we show where you can tell the viability is maintained even after 72 hours. Of course, along with viability, our product also helps maintain the cell composition and the gene expression. So this is data to show that that is not impacted by storing your sample in our preservation bar. So once you've found a way to store it and transport it for your experiment, if you're working with the tissue, another very important step is tissue dissociation. So here you want to get your tissue to a single cell suspension. So what are the main requirements to keep in mind? First is your single cell suspension should have high viability and good yield. You also want a method that can work on a broad range of tissue types, because your project might have different tissues and you don't want to be optimizing each one every time. You also want something that can work on a broad range of tissues amounts, because sometimes you might be working with biopsy samples, clinical samples that have very little. Sometimes you have a lot of sample to work with. So you need something that can work across the range. And of course, it's always good to have a short operating time protocol. You don't want the tissue to be sitting around for too long in the dissociating process, so in case your viability starts dropping. So for this, Singleron has a Python system. So this is tissue dissociation with walkaway automation. We have instruments that can either do 2 or 8 samples at a time. And so the way our protocol works is there is an enzymatic and a mechanical dissociation component to our tissue dissociation. So you can use our tissue dissociation kit, which is an enzyme that helps with breaking down the tissue, and along with that, use the mechanical dissociation, the Python or Python Junior, along with which comes a dissociation unit, which is a single-use consumable. And so our enzyme has been tested on more than 300 different tissue types, not just human, mouse as well. And here's some data just to show how our glioblastoma tissue dissociation was carried out. A quick comparison with homebrew methods that many customers use, competitor solution and our cell live solution. And as you can see, our cell viability is really high. Our mechanical dissociator, which is Python Junior or Python, has fully customizable plug-and-play suite of tested protocols. And what that means is we have a few parameters that are fully customizable by the researcher. You can play around with this and optimize it to work for your tissue type. And this instrument can deal with small sample sizes as low as 10 mg all the way to 4 grams. So it's really a broad range that it works with. Just showcasing some data, the three main things to keep in mind. Of course, you want cell high viability at the end of your dissociation, as you see here. Your data also needs to be consistent. So the transcriptome shouldn't get impacted every time you try to dissociate it. So data to show that it's consistent. And finally, you need to, your method should yield cell type representation that's consistent. And here you can see that you pick up the same cell types across the 3 samples. You also want a very easy method to do this, and we do believe our Python is quite simple to use. So you start off by opening this unit, you add a certain amount of solution and the tissue of interest that you want to dissociate, put the unit together, and then you use this, the Python to dissociate. So what usually happens when you put it in is the rotor helps rotate this plate, and so it's sort of a mechanical dissociation along with the enzyme that will break down the tissue. And so once you're done with this, you actually can collect the sample back and then do your downstream processing. So the basic downstream processing post this would be doing a quick QC where you need to figure out what is the cell count and the viability. So you want a good yield to carry on with your single cell workflow, and you want high viability. Usually we'd recommend above 80 to 85%. You also would want to do a cell size filtration, depending on the method. There's cell size filters that are used, 40 microns or 70 microns, to remove debris and any other undigested tissue bits. Finally, optional are red blood cell removal or debris removal. So these are also recommended before you do single cell sequencing. So I also want to point out that, of course, our Python Junior, along with the enzyme, can be used for single cell sequencing, but there are many other workflows that also this fits into. For example, if you want to do flow cytometry, any sort of cell sorting or a single cell culture, all of these require a tissue or a tumor sample to be dissociated into a single cell suspension. So these are also downstream applications for which the Python and the cell life solution has been used. So just to kind of show some data, this is data on different kinds of mouse tissue. And as mentioned, you want to look for high viability and good yield. So you can see here the viabilities are all in orange. It's all greater than 80%. And the cell yield is also quite high. Of course, as the tissue is different and the amount is different, you see that different amounts of cells are yielded at the end of dissociation, but all with very high viability. Finally, just to showcase some publications using these solutions. So here's a study that used it for mouse testes. So the application was to do single cell work, and so they've used the Singleron's tissue dissociation products for this. Similarly, this is for liver and brain metastats of breast cancer. The application is again single cell sequencing, and they've used our tissue dissociation. And finally, as mentioned, you can use the product not just for single cell workflow, but also for flow cytometry. So here's a study where breast tissue was dissociated with Singleron products and then used more for flow cytometry. Of course, the question that does come up is, do you really need to do automated dissociation? Lots of people might just do manual cutting up the tissue and grinding it. And so just to showcase some data, so this is comparing manual and Python Junior skin dissociation. What we do see is that when you do a automated method, your viability might, you may still be able to get high viable samples from manual, but the number of cells that you get are much higher when you use an automated method like Python Junior. And what that means is when you get more cells, you actually get more cell types and subtypes, especially the rare ones that you may be missing when you were doing manual. And all of this is with no compromise to gene expression profiles. So it is recommended that you actually do adopt an automated dissociation method. So moving down the workflow, we're next at cell partitioning and mRNA capture. So once you have a very good single cell suspension, you have to get going with capturing your cells and then the mRNA from individual cells. So what are the requirements when you choose a method? You want a method that can actually capture cells from different sample types and cell types. So there shouldn't be any bias in the capture to represent the actual sample. You want to make sure it can capture rare cell types, so that can be achieved if you are able to target more cells. Also capture sensitive cell populations. So during dissociation and just the general workflow, cell types that are very sensitive can either burst or die in the process. So you want to make sure they are not lost if you choose a method. And it would always be nice if you can actually QC any of these steps, so make sure that everything is going on track. And so here I'd like to introduce our single cell capture method. So the first part of our technology is the scope chip. So it is a chip that looks like what you're seeing in the image here. It's the size of a slide, so can be put under a microscope. And so these are the main key words that we would associate with it. A very gentle method of capturing, efficient because you can do between 500 and 30,000 cells on nuclei. Easy to get started with. As you can see here is sort of just a pipette to load the cells into the chip. And it can be, this whole process can also be streamlined and moved to an automated method, which I will mention soon. So if you put one of these chips under the microscope, what you'd see is that there are a whole bunch of wells on this chip. And this is where the cells will be captured individually. So we have three versions of this chip. The most common one used is the standard chip version. We have about 150,000 wells on this to capture up to 10,000 cells. The reason we have a lot of extra wells is to reduce the chances of doublets and multiplets. And so the size that these wells are about 40 microns. So that's sort of the limit. In many of the single cell technologies, you capture cells lower than this. If you're interested in capturing higher numbers, more than 10,000, we do have a high density version of the same chip where you can target up to 20 to 30,000 cells. We also have a large well chip, which is a customized product. And here the well size is 100 microns. So as I mentioned, if you do have cells sizes more than 40, there are not many options available to do single cell capture. We have one of that where we are actually able to capture larger cells in samples that don't fit the 40 micron cell size limit. So the second part of our technology would be barcoding beads. So these are beads that have unique cell barcodes, UMI or unique molecular identifiers, and the poly-T tail. So our profiling is done with, our mRNA profiling is usually done with poly-A, and that's where you have a poly-T which attaches to the transcript. So basically you need to get the cells and these beads onto this chip, and that's how the capture happens. So how do you do that? We have one way, which is the manual method of doing this. And so what you can do is pretty much load your cells and then the beads. So what you see in the video here are when the beads are loaded, it's very easy to do. As you can see, there's a pipette, a P200 pipette onto which you can use to just load the beads and cells. And then once you're done and all the lysis and capture happens, you use a magnet on top of this chip to pull out everything. So all of these steps mentioned here can manually be done. At every step, you can also put it under a microscope to check. So the first is if the empty chip would look with empty wells. Once you add the cells, you will notice about 10 to 15% coverage of cells. And finally, you add the beads to make sure all the different wells are covered. And once the lysis is carried out, you can pull all of it out with the magnet, as you just saw in the video. Of course, if you prefer a walkaway automation method for this, we have the Singleron Matrix Neo. So the instrument looks something like this, and the chip is slightly modified. So here, you just need to add all of the reagents at one point of time into the different reservoir or wells, and then it can actually do all of these steps for you. So you have an option of choosing. Here, you can run between 1 to 4 samples, and the runtime is about 38 minutes for the single cell capture. So The advantage here is that you can run different cell configurations chip configurations at the same time, and as mentioned, between 1 to 4. So if you have high throughput, this is a preferred method, and it will take care of all of these different steps for you. So our ~JEXScope kits, which are used for the single-cell RNA profiling, are very widely used. I've just pulled out some publications from our customers on various different tissue types. So this is on non-small cell lung cancer, so lung tissue, PBMC, human colon mucosa, or breast cancer. And as mentioned, our kits are also compatible with other species. So we've got publications, of course, mouse models. We've got one on insects, Drosophila, and then also on non-human primates. We also have on plants. So really our kits are compatible and are not dependent on what species you're working with. We also believe that we are able to push biological insights using our kits. So, for example, you have a chance to capture rare cell types if you capture a high number of cells. So here's a case where we use our high-density kit on a mouse embryo. So in a mouse embryo, as you can imagine, the immune cells are considered rare, so they're less than 0.15% of the different cells, but we are able to capture that as well, as you can see in this UMAP. And so it really pushes you to discover things about your data that you may miss if you didn't have a chance to target so many different cell numbers. Another sort of interesting facet is that we're able to capture sensitive cell types. So neutrophils are known to be very sensitive to change in environment. So they die pretty quickly during workflows and also have less amount of mRNA. So in cells like this can get missed if you're using a single cell capture method that stresses them out. Our workflow is microbial based, and so it's quite it doesn't stress the cells out. And so we have data here to show that you're able to capture a lot of neutrophils in this sample. So more than 40% was neutrophils, which are a sensitive cell type. And so this is something our customers find as well. So here's a publication that used our kits on pancreas tissue, and they were interested in the neutrophils in the sample. So they were able to capture that and look at gene signatures to further answer their research question. So I did mention earlier that you can also do single nuclei. And so this is possible if you have tissues that are already frozen. So there's some point you've actually collected these tissues and flash frozen them. So you can actually explore single cell or single nuclei on these. The things to keep in mind in that if you do single nuclei sequencing, you will pick up about 30 to 40% of the total mRNA. So it will be a little compromise on the amount of data that you will get. But having said that, it is useful data that you would otherwise not be processing or knowing if you didn't put it through the single nuclei pipeline. So just sort of a quick comparison, if you're working with immune cells, you are better off doing single cell work. But if you do have tissue types that are slightly difficult to dissociate or certain other tissue types, we do recommend considering single nuclei sequencing. So I will just bring up one or two examples here. So one tissue type is adipose. So these are fat cells that can float or are large or irregular in shape. And so in many technologies, they don't get captured very well. And so if this is a cell type of interest, then we sometimes do recommend trying single nuclei sequencing. Similarly, for large or irregular cells of interest from brain, muscle, etc. So these are, again, samples that, depending on the research question, you could choose to do single cell or single nuclei sequencing. So just to kind of highlight an example, this is using muscle tissue from mouse. If you did single cell sequencing, you would pick up all of these different cell types. So the immune cells, which if that is your question or research question of interest, you must do single cell. But if your research question of interest are muscle cell types like the myocytes, they may be lost in the single cell workflow just because of their shape, or because they're too sensitive to the process. So then you can consider doing single nuclei RNA-seq. And so this is, again, one of our customer publications. They work on muscle, which is frozen. They were frozen biopsy samples. So they chose to do single nuclei with our kits. And as you can see, they have picked up the myonuclei, which are the muscle cells of interest, including some immune cells at lesser numbers. And this has helped them answer their research question. So I've been talking about mRNA profiling, our ~JEXscope kits that can help you profile the mRNA. But we do have many other multi-omic kits in our portfolio that help add an extra layer of information along with the mRNA. So the few that I would focus on today are the immunoprofiling. If you're interested in understanding the immune profiles of different cell types, in different cell types, it's here. And then we also have a focus scope kit, which I will cover after. So moving to immune profiling, just to kind of show you what are the different cell types that when we talk about immune profiling, we're looking at these innate and adaptive immunity cell types. So if you took a sample and you did single cell RNA sequencing, you can identify all these different cell types, either from a tissue sample or from a whole blood sample. So once you've identified all of this, there is a lot of research interest in identifying or understanding specifically cell types like T cells and B cells as they express receptors on their surface for antigen recognition. So in the field of immunology, this becomes extremely important, getting more information on these cell types and information on the receptors on T and B cells. And they're commonly known as TCR and BCR. And so this is possible by actually profiling something known as VDJ sequences on these receptors. And so what people would be looking for is to actually profile these at the single cell level. And so for this, we have the ~JEXscope VDJ kit. So we profile both the transcriptome as well as the VDJ sequence from individual cells. How do we do that? So if you recall, I did show you a structure of our bead, which has poly-DT to capture the mRNA. What we do is we tweak some oligos on that bead to have probes that capture close to a CDR3 region on the immunoreceptor. So CDR3 region is considered the most diverse part of the receptor, and so most researchers are interested in this region. So we're able to pull that out at the single cell level with really strong VDJ specificity. This is just some data to quickly showcase how our kit, when compared to market a competitor, we're able to capture up to 83% of the cells having both the alpha beta chain. So these are part of the TCR sequences, and then also a lot of the high number of VDJ transcripts from these cells. So the main takeaway is that it enables the CDR3 detection, which helps you do clonotyping of these cells. So as mentioned, we have a kit to do the CDR3, but in case the researcher is interested in getting more, that is the full-length TCR-BCR, which includes regions such as CDR1 and 2, we also have a kit for that. So here we would call it the full-length immunoreceptor profiling. And we have some data here. I just want to showcase and highlight that using our kit, you get a large number of cells that have productive VDJ pairing span, which pretty much means that there are a lot of cells that give you the full-length immunoreceptor and a lot of reads that get mapped to the VDJ gene. So this is for researchers that are interested in profiling the full TCR or BCR sequence, and you can also do clonotype. And so just to kind of put the two kits side by side, what we offer with the ~JEXscope VDJ kit and the immunoreceptor full length, the applications can be slightly different. So if you're interested in just clonotypes, understanding the diversity of the TCR, BCR, tracking immune responses, you use the VDJ kit. If you're more into personalizing your immunotherapy, so you need to identify these cells and then do some kind of cloning to for that reason, you will need the entire sequence. So then we recommend you do the ㍍Circle kit. So here's sort of a customer example where they have actually profiled the tumor microenvironment, but here they were interested in the immune profiling. So they do single cell RNA-seq. The samples are taken and RNA-seq is run. You identify the different cell types. So you can see here, they've identified all the different immune cells using the RNA. And then using the TCR part of the kit, they were able to actually get the clonotypes and the TCR repertoire. The different clonotypes, you can see what are the more common or dominant ones. And between the different samples, they see differences in this TCR repertoire, giving them very important insights into their data. The next kind of kit that I would focus on is Focus Scope. This is probably one of the unique solutions that we have. So here we're able to profile at the single cell level the transcriptome, along with targeted variant detection. So what do I mean by this? If you look at our beads, that structure that I showed you before, we're able to capture the mRNA with the poly-DT, but we're also able to tweak these beads to add target regions. So for example, you can detect single nucleotide changes, alterations, mutations, or gene fusions at the single cell level. We're able to put those probes on these beads and capture it. We're also able to do non-poly A transcripts, example, viral transcripts. So not all transcripts may have the poly A tail. And for example, viral transcripts don't have this. So if you're interested in viral infection in cells, you will not be able to capture it with standard mRNA capture, which are using poly-DT, but we're able to do that with this particular kit. So what we have are pre-validated panels. We have 4 that we offer based on different sort of target regions. So we have one for blood cancer, which include gene fusion markers of interest, one for lung cancer, one for clonal hematopoiesis, and then one for EBV, that's Epstein-Barr virus. So these are panel or kits that you can just buy off the shelf, and each of these kits will give you the RNA, the transcriptome, and the target panel of interest at the single cell level. What we're also able to provide is if none of these kits match your interest, but you have targeted regions that you'd like to put together, we are able to offer customized panel. So we can help you, you can provide us with the markers, and then we will actually design the kits for you, and then you can go ahead and use these kits. So here is an example of one of our customer publications where they were interested in profiling hematopoietic stem cells, looking at the viral profile of these cells. And so they were able to use the single cell part to annotate all the different cell types, and then use the EBV layer to actually understand which of these cell types have EBV infection. And then they're able to see how this has progressed between the different cell types and do more analysis on that. Okay, so right now we've kind of moved down the workflow. We've figured out how to dissociate our samples. How do you capture the different cells? What are the kits you can use to capture mRNA or any other additional information? So now we're at sequencing. All of our kits have everything required to give you a library that's ready to sequence. It's tailored to Illumina sequencing, but you can use our libraries by tweaking them to run on any other long or short read platform. Yeah. Finally, once you send it for sequencing, you get back all this data, and then you need to find a way to actually address the questions that you started with. We have a whole suite of products available, and I'll just go through these in short. So the first one is CellScope. So from a sequence provider, you would get your FASTQ sequence data. For pre-processing that, you can use CellScope. So this is free to download. It's on GitHub. You take the data, you run it through the workflow, and what you would get is your count matrix. So this is the first main result from a single cell pipeline, the different number of cells that you have targeted, and gene counts for the different genes that were detected in the data. So you get one of these count matrices, and this process is done by taking the data, mapping it to the reference genome. So depending on if you're working on human or mouse or any other organisms, you map it, and then you get the and then you get it into this count matrix. And using this count matrix, you answer your different questions. So the most common sort of analysis that you would see in a single cell paper would be a visualization in terms of a UMAP. I've been showing a few of these. So the different cells that are closer together means that their transcriptomes are more similar compared to something that's further away. Just in case I didn't mention, each of these dots are a single cell. You can then go in and annotate and give names to these, and then you do all of these usual standard analysis, differential gene expression, pathway, cell-cell interaction, etc. Of course, you can do this all on your own, but we have also tools that empower you to do code-free data exploration. And so this is sort of a whole suite of tools that we provide. We have a few that are online. One is a single-cell sequencing database. So this is very useful for researchers who are really looking to browse clinical data sets and look for markers and then kind of compare their own data. We have this curated, manually curated with cell type annotations and maintained for people to log in and use this database. We also have a standalone cloud-based data analysis platform that actually has come out this week. You're able to actually upload your data, do integration of data sets, automated analysis, and a lot of the customized analysis that I was just talking about. And finally, we also have a software that can help you visualize your data. That's a local installation. So some of these are free to use, some are subscription-based models. So for the last part, I think I'm just going to sort of bring to your attention some of the advantages that you do get by using the Singleron workflow. We do believe that we capture end-to-end, so you have the entire value chain really from how to prepare your samples and to analyze your data, and then a number of unique multi-omic solutions. Of course, the question then comes up, how does it compare to other technologies that you may have heard about? So I will just do a quick overview. One very popular technology is droplet-based technologies. And so here you actually get your cells and the reagents and the beads into a droplet using oil. And so this is sort of the advantages that we do feel we have is that for our technology, you don't really need to have a sophisticated instrument to do this. You can choose to use an instrument just to have walk-away time, but it's not necessary. And because there is no vacuum involved, just putting the cells onto a microwell chip, there is no issues of clogging or using force to get cells into droplets. So sensitive cells also perform better, or irregular cells also do better using our technology. And again, we have some multi-omic solutions that are quite unique. Of course, just to showcase some data here to show when compared to supplier X, which is a droplet-based technology, we're able to capture quite a number of cells because we have an option to do a manual, a high-density chip. The mean reads and UMI are all very comparable, and we also get more higher median genes per cell. Just some QC metrics, again, showing that the samples run with our technology are very comparable in terms of QC metrics, even better. And of course, the most important thing is, are you picking up the same cell types? And we do see that we pick up the cell types, but I also want to just draw your attention to the neutrophils in purple here. As you can see, when you do Singleron technology, you do get more neutrophils when compared to droplet-based methods. So the main takeaway also is that if you actually wanted to integrate or compare with 10x, we do, or a droplet-based technology, we find that all of the data results that you get, for example, the top genes of interest are the same, which is the most important thing to take away from this. So you can either replace your experiment using our method, or you can also integrate if you want to, if you happen to have data sets from our technology and any other legacy data. And this is the same that we see when we use our BCR, TCR, BDJ kits. So here's just some data comparing droplet-based technology with ours. We identify more BCR and TCR clonotypes as well. So all in all, the takeaway is that we are able to sort of give you the same quality of data at a more cost-efficient manner. The other technology that's popularly sort of heard is combinatorial barcoding, indexing, or the split-seq method. And so here are some advantages that we feel our technology has when compared to this. For us, you can actually start with limited starting materials. You don't need millions of cells to actually do the workflow. So now amount of starting material is not limiting with Singleron. You also have the flexibility to do one sample or multiple samples. You don't need to start your project with a lot of samples to make it cost efficient if you're doing single-run methods. We are also quite easy in terms of workflow, 1 to 2 days, and you can go from sample to your library. So it's not a long drawn protocol over 2-3 days to actually do the pooling, splitting, etc. And then again, to point out multi-omic solutions that are quite unique to our technology. So I'm just going to end with a few examples where we really show you that you can go sample to answer. So here's a study where researchers were interested in looking at the immune cells from HCC samples. And so here they were able to take the liver samples, do preservation of the samples after dissecting from the mouse using our cell life preservation buffer, dissociating it using our dissociation solution, and then doing the single cell workflow using the ~Code kit. Similarly, I'm also going to just point out this study. So here it's on organoids, again, another model that's used quite popular right now in a lot of different fields. So here you can use our technology was used to dissociate the organoid and then do single cell sequencing to answer. And they were studying a mitochondrial disease stemming from POLG mutations. I'm also going to just flash this webinar. So we're actually running a single cell innovative webinar series. And here is one of our customers who, just as I mentioned, has used our technology on midbrain organoids. So it is a bit late in local time, but if you're interested, please do register and you can listen to it. And finally, I'm open to any questions and any other single cell requirements that you have. Singleron is really happy to support on that. And of course, you can also reach out to Le Korea for any questions that you have or anything else related to single cell and the other products that they have on their portfolio. I will end here, and I'm open to questions.